look how adorable this fish? Look at their pattern. Yes, it's called zebrafish. But have you ever imagined how they develop until grow that big and can swim in the water freely? If you want to know more about the zebrafish, let us explain it to you. The Nugroho, commonly known as zebrafish, is a tropical freshwater fish belonging to the minnow family. It has become another popular model organism to study fundamental biological questions. It is a small 1 to 1.5 inches fish that grows easily in aquaria.
Let's dive to the taxonomy part of zebrafish. Zebrafish is in the kingdom of Animalia. Zebrafish have species name of Rario and genus name of Danio. It belongs to the family of Cyprinidae under the order of Cypriniformes in the class of Actinopterygii with the phylum of Chordata.
Now, let us explore about the history of zebrafish development. The use of zebrafish as a model organism was pioneered at the University of Oregon, USA by George Streisinger in 1970. He is the founder-father of zebrafish developmental and genetic research. In 1990, the first large-scale mutagenesis of zebrafish were conducted by Christiane Nusslein-Volhard in Oxford University, United Kingdom, to identify developmental mutations. Thomas Luke of Dana-Farber Cancer Institute, Boston, uses the translucent zebrafish to study how cancer behaves in 1995.
Here are the zebrafish anatomy.
Zebrafish cycle starts after fertilization. Followed with cleavage stages where cell divisions occur in precise planes of orientation. Next is the gastrulation stage, where the embryo is in relation with both animal and vegetal pole. After that, organogenesis stage takes place. Then it hatches as a free swimming feeding larvae. Lastly, it reaches sexual maturity around 3 months of age and can live up to 5 years. Zebrafish eggs are fertilized externally, and they're also called egg scatterer. The female will find a comfortable place for them to spawn their eggs, and the male will also be ready for ovulation. Cell divisions occur in specific orientation planes during early cleavage stages, and the length of the cell cycle is relatively synchronous and have a uniform length of time. However, the length of the cell cycle changes and becomes asynchronous and non-uniform during the mid-blastula stages. Now, the process of gastrulation requires dramatic cell movement and that the cells must involute to form mesoderm. The leading edge of the involuting mesoderm, known as the shield in zebrafish, and then measured or noted that cell movements drive the involution forward. The process of gastrulation is morphologically the earliest time point at which the anterior and posterior axis of the embryos can be defined.
Gastrulation and formation of germ layers, cell of epiblast and hypoblast. It intercalate on the dorsal side. It forms localized thickening with involuting cells. It has embryonic shield, and this is the functional blastopore lip of amphibians. blastoderm margin converges anterior and dorsally towards embryonic shield. Movement forms chordomastoderm, precursor of notochord. And now is the segmentation period of the zebrafish. Duration of this period is from 10 to 24 hours. It is the transition from 5 somites to prim 5. Dermis, vertebrae, and skeletal muscles are formed. This is also the beginning of primary organs development. Embryo elongates and the tail bud at the caudal end becomes more prominent. The first body movement occurs and the notochord is still forming.
Now, I will explain about model organism. A model organism is a non-human species that has been widely studied, usually because it is easy to maintain and breed in a laboratory setting and has particular experimental advantages. They are used in the laboratory to help scientists understand biological processes.
This is the advantages of zebrafish as a model organism. It is optically translucent embryos. It has rapid hatching of eggs. The maintenance cost is significantly lower than those for mammals. It is amenable for molecular and genetic analysis. As zebrafish eggs are fertilized and develop outside the mother's body, it is an ideal model organism for studying early development.
The reasons why zebrafish are used as a model organism from the genetic aspects are: they can be cloned from somatic cells, they can be made transgenic. Also, 70% of protein-coding human genes are related to genes found in the zebrafish, and 84% of genes known to be associated with human disease have a zebrafish counterpart. And lastly, as a vertebrate, the zebrafish has the same major organs and tissues as humans. Their muscle, blood, kidney, and eyes share many features with human systems.
Zebrafish is used as an animal model for research study. The study includes regeneration of heart, tail regeneration, retinal regeneration, human pigmentation, cancer research, and also autism.
Mammals respond to a myocardial infarction by irreversible scar formation. By contrast, the zebrafish are able to resolve the scar and to regenerate functional cardiac muscle. The reparative and regenerative process is achieved through SOMAD3 dependent TGF-beta signaling. During a heart attack, heart muscle cells are deprived of oxygen and they die, leaving scar tissue. Scientists are working to find out the specific factors involved in this process to see if this will help us to develop ways of repairing the heart in humans with heart failure or who have suffered heart attacks.
Tail fin regeneration. First, zebrafish fins are complex appendages that quickly and reliably regenerate after amputation, restoring both size and shape. Secondly, the key regenerative units are their many rays of dermal bone, which are segmented and lined by osteoblasts. Thirdly, an amputated fin ray is covered within the first several hours by epidermis, and within 1 to 2 days, a regeneration blastema forms. The blastema is a proliferative mass of morphologically similar cells formed through disorganization and distal migration of fibroblasts and osteoblasts. Fourth, blastema formation is the only one step in zebrafish tail fin regeneration. Fifth, WNT signaling positively regulate blastemal proliferation and outgrowth.
Zebrafish also can be used in human pigmentation research. At embryonic and early larva stage, the neural crest cells get differentiated into three cells, displaying alternating stripes. The first one is melanocyte, represent the blue color, and the second one is iridophores, represent the silver color, and the last one is xanthopores, represent the yellow color. Further study in zebrafish pigment cells is essential since they provide many opportunities to learn the nature of human skin color.
Retinal regeneration. The researcher studied Mueller glial cells in the eyes of humans aged from 18 months to 91 years old, and were able to develop them into all types of retinal neurons. The stem cell of zebrafish successfully migrated into diseased rats' retinas and took on the characteristic of the surrounding neurons. The team stated that they intend to develop the same approach in human. Stem cells from zebrafish, the staple of genetic research, could regenerate damaged cones in retinas and restore eyesight to people.
This is a cancer cell. In cancer research, zebrafish have been used to make several transgenic models of cancer, including melanoma, leukemia, pancreatic cancer, and colon cancer. Researchers have created a model of cancer in zebrafish that allows them to capture live images of tumors forming and growing.
Zebrafish model gives new insight on autism spectrum disorder. A team of researchers at Massachusetts Institute of Technology focused on genes that have been found to be either missing or copied in about 1% of patients with autism. Their study revealed that when they deleted these genes from zebrafish embryos, nearly all the fish developed brain abnormalities. It is found that estrogens, the primary female sex hormone, could reverse abnormal behavior in zebrafish carrying a mutation in CNTNAP2, a gene linked to genes of autism in humans, SYNGAP1 and SHANK3. The researchers found that zebrafish carrying the CNTNAP2 mutation are more prone to seizures than zebrafish without the mutation.
Now, what would be the future prospects of this study? Well, as zebrafish are now being used in virtually all disciplines, from neurogenesis to oncogenesis, from behavior to genetics, its popularity is steadily increasing. The emergence of zebrafish conferences and the increasing presence of zebrafish studies at large annual meetings are also boosting the use of zebrafish as an appropriate model organism.